Hello everyone, welcome back to my channel. I am Vamshik Shetty, pursuing second year B in Sri Venkateswara College of Engineering, Bangalore. Today we are going to discuss about the major topic in the biology for engineers, that is DNA origami and biocomputing. The main agenda is to discuss about in this video is that is DNA origami and some of the procedures of the DNA origami and some of the major applications of the DNA origami. Next, the biocomputing and procedures of the biocomputing. What are all the procedures that are undergoing in the biocomputing process and some of the major applications of the biocomputing. Let's start the video. Let's first let us discuss about the DNA origami. DNA origami is the nanoscale folding of DNA to create arbitrary 2 and 3 dimensional shapes at the nanoscale. The specificity of the interactions between the complementary base pair makes DNA a useful construction material through design of its base sequence. DNA is well understood material that is suitable for creating scaffolds that hold other molecules in place or to create structure on all its own. The procedures of the DNA origami is that to produce a desired shape, images are drawn with a raster fill of single long DNA molecule. This design is then fed into the computer program that calculates the placement of the individual staple strands. Each staple binds to a specific region of the DNA template, and thus, due to Watson-Crick base pairing, the necessary sequence of all the staple strands are known as are known and displayed. The DNA is mixed, then heated and cooled. As the DNA cools, the various staples pull the long strand into the desired shape. Designs are directly observable via several methods, including the electron microscopy, atomic force microscopy, or fluorescence microscopy when DNA is coupled to fluorescent materials. In this procedure, we are going to learn about the how the DNA is extracted. That is by using the DNA template using the Watson-Crick base pairing. First, we are going the DNA is mixed and then heated and cooled. As the DNA cools, the various staples of the DNA is obtained, and thus the DNA is finally obtained by this procedure of the DNA origami. And the bottom-up self-assembly methods are considered promising alternatives that offer cheap parallel synthesis of nanostructure under relatively mild conditions. Since the creation of this method, software was developed to assist the process using the CAD software. This allows researchers to use a computer to determine the way to create the correct staples needed to form a certain shape. One such software called CAD DNA is an open source software for creating such structures from the DNA. The use of this software has not only increased the ease of the process but has also drastically reduced the errors made by manual calculations. Here the typical diagram of the DNA origami formation is given here, and the crossover representation also given here. The 2D and the 3D architecture of the DNA is also given here. You can see that the DNA origami formation can be takes place. That is, the staples and the scaffolders combined to form the scaffold folding. And let's move on to the some of the applications of the DNA origami. That is, first we are going to discuss about the nanoelectronics. In the field of nanoelectronics, DNA origami has shown promise in the field of nanoelectronics by serving as a scaffold to arrange the nanoscale components, such as carbon nanotubes or quantum dots, into specific patterns and configurations. This could lead to development of smaller, faster, and more efficient electronic devices. This is the application in the nanoelectronics. And next, the drug delivery. The DNA origami nanostructure can be designed to carry drug molecules or therapeutic agents to specific targets in the body. The nanostructure can be functionalized with the ligands that recognize specific cells, enabling the targeted drug delivery and reducing the side effects. The next applications of the DNA origami is the biological sensing. DNA origami based biosensor can be constructed to detect the specific molecules such as proteins, enzymes, or even small molecules. The precise arrangement of the DNA strands allows for highly sensitivity and selectivity in detecting target analytes. Next, the nanorobotics. The DNA origami has the potential to serve as the foundation for the building nanoscale robots. These nanorobots could be designed to perform tasks at molecular level, such as manipulating cells or delivering payloads with higher precisions. The next is the nanophotonics. The DNA origami can be used to arrange the chromospores and the other photonic elements to create a new types of nanophotonic devices such as light harvesting structures or the sensors. These are the some of the applications of the DNA origami. Let's move on to the next topic, that is biocomputing. A computer that uses a components of the biological origin such as molecules of DNA instead of the electrical components is called as biocomputing. The device is rudimentary, it can be only perform basic high school level math problems. In a quest to understand and the model the healthy or the sick human body, researchers and medical doctors are utilizing more and more quantitative tools and techniques. This trend is pushing the envelope of the new field we call biomedical computing as an exciting frontier among the signal processing, pattern recognition, optimization, nonlinear dynamics, computer science, and biology, chemistry, and medicine. The biocomputing is a process, a computer that uses the components of the biological origin, that is the DNA molecule, instead of the electrical components. Let's move on to the procedures that is involved in the biocomputing. The process of biocomputation typically involves the following steps. The first step is the encoding, where the in the encoding process the information or the computational problem is encoded into DNA sequences. Each DNA strand represents a specific piece of data or an instruction. And the second step of procedure is the manipulation. The DNA molecules are manipulated and processed using the biochemical reactions. These reactions might involve the hybridization, ligation, cleavage, or the other enzymatic activities. Let's move on to the next step, that is the computation process. As the biochemical reaction occurs, the DNA molecules interact and undergo specific transformation that encode the computational steps needed to solve the problem. The final step is to detect the DNA by using the biocomputing process. That is, the final result of the computation is read out by analyzing the output DNA sequences. This may involve using the techniques such as gel electrophoresis or sequencing. The process of the biocomputing is given in the typical diagram, where the DNA metabolism and the DNA computation and the detection process are all given here in the uh, in a in a typical diagram. Let's move on to the some of the applications of the biocomputing, where the first application is the data storage. DNA has an incredibly high information density, making it an attractive candidate for the data storage. Biocomputing could pave the way for ultra compact and long term data storage solutions. DNA based data storage has the potential to store the vast amount of data in a small volume, overcoming the limitations of traditional data storage technologies. The another applications of the biocomputing is the molecular diagnostics. Biocomputing can be used to design molecular level diagnostic tools that can detect specific DNA or RNA sequences associated with the disease or genetic variations. This can lead to highly sensitive and specific diagnostic tests for various medical conditions. And the next application of the biocomputing is the molecular robotics. Biocomputing has the potential to contribute to the development of the molecular scale robots that can perform tasks at the nanoscale, such as targeted drug delivery, cell manipulation, or the environmental remediation. Another applications is the optimization and the pattern recognition. DNA computing can be applied to solve the complex optimization problems such as routing, scheduling, or resource allocation. Additionally, DNA based pattern recognition systems can be developed for various applications, including the image processing and the data analysis. And at last, the last final applications of the biocompu- biocomputing is the cryptography. DNA based encryption methods have been explored as an alternative to traditional cryptographic algorithms. DNA based encryption offers the advantage of high information density and the potential resistance to the certain types of attacks. So these are the overall applications of the biocomputing. Yeah, that's it for this video. Thank you. For more videos, please like this video, share this video and subscribe my channel. Thank you.